Calvin cycle's purpose is to convert carbon dioxide into carbohydrates or sugars, specifically glucose for plants. This process occurs in the stroma of the chloroplast, unlike the light reactions which occur in the thylakoids. The first stage is carbon fixation, which captures carbon dioxide and attaches it to the 5-carbon molecule RUBP through the enzyme ribisco. This creates a 6-carbon molecule that is soon split by water into 6 3-carbon molecules known as PGA. In the second stage, reduction of energy sources occurs so they can fuel the process. ATP is reduced to ADP due to its donation of a phosphate group to each organic molecule. NADPH is reduced to NADP plus through its donation. The reduction of NADPH converts the PGA molecules into a molecule known as G3P. The 6 molecules of G3P becomes 5 in order for one to be used by the cell to make glucose and the rest to regenerate RUBP. In the last cycle of the Calvin cycle, RUBP must be regenerated in order to fuel the next. The 5 G3P that were recycled donate their carbon molecules for a total of 15 donated. 3 more ATP is used and is also reduced to ADP. 3 new RUBP molecules are created because they require 5 each.
Here we will depict the light-dependent reaction of photosynthesis. This is when the water molecule splits. Shown here is a water molecule consisting of 2 hydrogen atoms and 1 oxygen atom, held together by weak hydrogen bonds.
When the light enters the thylakoid and hits the H2O molecule, the oxygen oxidizes and goes into the atmosphere.
The hydrogen then becomes hydrogen ions that move on to the electron transport chain.
So on photosystem 2, the light energy is absorbed by my pigments and passed pigment to pigment until it reaches my reaction center called P680. And then once I receive an electron that travels through the chain, I'm going to energize it with that light energy, and then I'm going to pass it on to the acceptor molecule.
The acceptor molecule passes the electron to photosystem 1 but loses energy as it does so, creating a concentration gradient. The H ions are passed from the stroma to the thylakoid through the gradient.
So now that the H ions are in the thylakoid, they go to ATP synthesis.
They drop down into ATP synthase to make ATP.
So then ATP is produced.
Back to the electron transport chain. Photosystem 1 is hit with light. It is passed pigment to pigment to reach the P700 center. I then receive an electron from the acceptor molecule, and I excite the electron using the light energy, replacing the energy that the electron lost traveling from photosystem 2. I pass the electron back to the acceptor molecule.
Okay, and then the electron travels down the second part of the chain, which is shorter, and once it reaches the end, it's dropped off to an NADP plus molecule, and then it waits for a second electron to come through the chain, and then the NADP plus is reduced to NADPH.
So the products of the electron transport chain are the NADPH and ATP.
And that energy provides the cell, like the energy it needs to go through photosynthesis, and photosynthesis is important because it provides the oxygen that we all need to survive.
Glucose is important.